Hello everybody, and welcome back to General Biology 1. Today we're going to talk about cells and what happens when they get too big and they need to divide. So this is a cell undergoing the process of division. It's actually part of development. We're not going to talk specifically about development today, but we're going to talk about the two processes that allow cells to divide and multiply. So the first question is, why are cells dividing in the first place? When does this occur? What are the factors that are going to dictate that? Well, every cell, just about every cell, will typically divide at some point, right? And usually undergo multiple divisions. And that's something that they do. And the other thing they do is they grow. And when a cell grows, it grows in volume. And if you recall, volume increases by a factor of 3. But the surface area, the membrane itself, because it's a surface area, increases by a factor of 2. Meaning at some point you reach a volume that cannot be sustained by the surface area of a cell. If you recall, diffusion, osmosis, all of that is happening through the cell membrane. So when a cell gets big enough, it needs to divide. So it can decrease its volume to surface area ratio. And this process is known as mitosis. And the goal of mitosis is to produce two cells, two daughter cells from the parent cell that are essentially genetically identical. You're just cloning yourself, basically. So these are some terms that you will be required to know the definitions of, and we'll kind of work our way through them. But they're both genetic terms as well as terms describing particular aspects of mitosis as well as meiosis, which we'll get to later. So these are important terms to know, so be on the lookout as we move through this, right? So a cell's life cycle, it looks like this. Once it's produced, usually through a process of cell division itself, cytoplasm starts to accumulate, organelles get more organized, membrane gets more responsive to whatever the environmental conditions are, and it starts to grow. And that's the G1 phase of something called interphase, which is essentially most of the cell's life when it's not dividing. And then once it reaches a certain point, usually because the size of the cell is too big, it will start the process of replicating its DNA, right? DNA synthesis. So it'll enter into the S phase of interphase. And then once DNA is duplicated, it's going to undergo another growth period, right? This is the G2 phase of the cell cycle. And then the process of mitosis will begin, where the DNA that has been duplicated is now going to be divided among two cells. And following immediately after the process of mitosis, which is the division of the genetic part of the cell, you're also going to get a physical separation of organelles, cytoplasm, things like that, and that part is called cytokinesis, which for the most part they overlap quite heavily. So that's the M phase, or the mitotic phase. The end result is one cell will produce two daughter cells that are just a bit smaller, but each with the full complement of DNA that the parent had. So, a couple quick things, some terms you want to clean up. DNA is housed in chromosomes, and we'll get to a little bit about how that is organized soon. But in a diploid organism, every cell has homologous chromosomes, which means that one is derived from the maternal lineage, from the mom, one's derived from the paternal lineage, from the dad. So these two chromosomes are essentially the same chromosome, meaning that they have the same genes that code for the same things, the same proteins on them. They're located in the same places on the gene or the locus. And they might have different versions of the gene, in other words, alleles. So Homologous chromosomes in a diploid organism, one comes from each parent. They have the same genes, but maybe have different alleles for them. So it's important to remember this as we move through. And it's often a source of confusion. A chromosome is DNA that has been organized in preparation for cell division. We think of a chromosome as being this structure that has an X and is held together in the middle by a centromere, which is a protein, that kind of protein complex that holds them together. The chromosome is the genetic unit. But when you see that X, what you're really looking at is two chromosomes that are identical copies of each other. And these chromosomes that are copies of each other are known as chromatids or sister chromatids. So you see the X after the S phase has happened. You see the X after the DNA has already replicated. So what you're looking at is chromatids, sister chromatids, copies of each other, copies of the same chromosome. So this is something that's often a source of confusion. So it's important to keep these two things straight. The difference between a chromosome and a chromatid. Chromatids are copies of the chromosome, and they're usually held together. And what chromosome is, like I mentioned, it's DNA. DNA, double helix, you all know that. This double helix is wound around nucleosomes, and they form these little structures, these nucleosomes, which then kind of join together and form bigger structures, histones, which are then organized as chromatin. And most of the time, when the DNA is in the somatic DNA is going to be in the nucleus of the cell. And inside the nucleus of a cell, it's usually just this long spaghetti string of chromatin. It's only after cell division has occurred, and only after, or right before mitosis is to occur, that's when you see the chromosome fully condense. And we'll get to a little bit about what stage that is. So it's important to remember, a chromosome is what you can see, but all you're looking at is really, really compacted DNA. And it's quite wonderful if you think about it. Okay, so what happens during mitosis? Well, chromosome duplication happens during mitosis. A chromosome is inside the cell. A chromosome, again, is a DNA molecule. That DNA molecule replicates itself and forms another copy. So you have these two copies of each other, the sister chromatids. Again, this is the thing that looks like the X. And then what's going to happen during the process of mitosis is it's going to then divide the chromatids, and each resulting daughter cell is going to have one of the two sister chromatids. Right? So they're going to be separated. And if you look at the picture, the parent cell, number 1, looks the same as number 3 cells, right? And each of those now has one copy of the DNA molecule. So the goal behind mitosis is to have chromosome duplication and then a splitting of that duplicated chromosome. So each cell ends up with the full complement of DNA that the parent cell had. Okay, so the stages of mitosis. Let's do this. Remember, DNA has already been replicated and the cell has gotten big enough, and now it's going to undergo mitosis. And during that G2 stage of the cell life cycle, during interphase, what you're going to do is you're going to start to see the nuclear envelope start to dissolve. And the DNA is going to start to condense, and eventually it will condense into the chromosomes. So at the same time, you have these centriole pairs, these little sort of microtubule factories that are going to start to make their way to the opposite ends of the cell, the poles, if you will. So think of like North Pole, South Pole. And by prophase, the first stage of mitosis, right, interphase is its own thing. By prophase, you now have fully condensed chromosomes. They have their sister chromatid, right? So they're paired up with a centromere in the middle of them. And at the same time, you have these centrioles moving out to the poles that are starting to form these long microtubules it's headed towards the center of the cell, right? So that's called the mitotic spindle. And seeing all these microtubules comes out kind of gives it a star shape, so they call this the aster. So think like asteroid, right? So astro. And by the end of prophase, the nuclear envelope is completely gone. You now have fully formed chromosomes. They look like the X with the sister chromatids, and the centromere is going to attach to the microtubule at the kinetochore. And the kinetochore is basically where they meet, and there's a little interaction that happens there. And by the end of it, by late prophase, you're ready to divide. Note here it says prometaphase. I've always learned it as just the later stages of prophase. I'll leave it at that. Now, metaphase is important because if you're going to learn anything at all, learn metaphase. It will allow you to tell if a cell is undergoing mitosis or if it's undergoing meiosis. Meiosis has two divisions, meiosis 1 or meiosis 2. The only way you can tell them apart is by understanding metaphase. So if you learn metaphase, you could reverse engineer the rest of the stages. So in metaphase, the chromosomes are now all lined up on the middle. So think of like the equator, and you've got the asters on either side, right? And you have the all of them attached to the mitotic spindles. Notice how they're attached, right? The chromosomes are basically split evenly across the whole spot. So here in this cell, in this example, there are 6 chromosomes. Each of these 6 chromosomes are attached to the middle. That's metaphase. It's going to be different in meiosis 1 and meiosis 2. Now what's going to happen is that centromere is going to kind of come apart, and the mitotic spindle is going to start to pull, quote unquote, pull the chromatids to either end of the poles. So now you have DNA, the same DNA, being pulled to 2 separate poles, right? So that's anaphase, is the actual separation part. And then by the end of it, you end up with 2 cells that each have the full complement of DNA that the parent cell had. And now, at the end of anaphase, you enter into telophase. So what's happening in telophase? Well, the nucleus is starting to come back. The nucleolus, which is a small part inside the nucleus, is starting to form. The nuclear envelope is starting to surround the chromosomes, and those chromosomes are decondensing into the chromatin. At the same time, you're basically forming 2 nuclei. At the same time, you're also dividing the cells in terms of their cytoplasm. You're forming a new plasma membrane that's going to then separate the cells completely. That process is known as cytokinesis. So telophase is the genetic part with the chromosomes. Cytokinesis is all the other stuff. So these things are happening at the same time, and the end result is 2 daughter cells with the exact amount of DNA that the parent cell had. Right, so they're basically 2 daughter clone cells. So now you want to understand cell division. Well, there's some steps you can take, and it's good to kind of back away and look at this from a more broad perspective. So first of all, if you're interested in the chromosomes, one thing to do is to count the centromeres. Right? If you have 6 chromosomes during mitosis, well, then count those centromeres. And I'll tell you that you should have 6 of them. Right? And the other thing to remember is chromosomes are just DNA. It's condensed chromatin. And synthesis happened earlier. So you have 2 copies of the chromosomes. Right? You have one from the parent, one from the mom, one from the dad. Right? Those 2 copies, those versions, are homologous. So we say they're homologous chromosomes, or a pair of homologous chromosomes. Now, after DNA duplication, a chromosome will produce its copy, its clone, and that's when it becomes a sister chromatid. So homologous chromosomes are, each chromosome has the male version, the paternal version, and the female version, right? The maternal version. So those 2 are going to be homologous chromosomes, but each of those will divide and produce its own copy, right? So you'll have the maternal chromosome producing its copy and the paternal version producing its copy. So those will be the chromatids, the sister chromatids. The other thing to remember is on chromosomes there are genes. And when you have two homologous chromosome pairs, or a pair of homologous chromosomes, one from either maternal or the paternal, those different versions of the gene, the alleles, they're found on those different chromosomes. So it kind of gives you an idea of physically where these things are located. The other thing to remember is all of the genetics put together is that other term that we use, the genome. That's the full range of DNA for the organism, all the chromosomes being taken into account together. So understanding these steps will help you kind of understand what's what, because the terminology does get confusing. Chromosomes, chromatid, chromatin, I get it. So take a look at it, see if you can make sense of it. Now, humans have 22 pairs of autosomes and a pair of sex chromosomes. So this is an example of a karyotype for a human, right, where you have the homologous chromosomes lined up with each other. Now, just so we're clear, here you have the chromosomes, and each of these pairs, one is from the mom and one is from the dad. Notice there are no centromeres, right? The centromeres will come when each of those strands of chromosome produce its complement, right? Its copy. That's when you see the centromeres. That's when you see the X. You don't see the X here. Some of these look like they could become X's, but they don't, right? In the last pair, the XY chromosome, well, you see an X and a Y chromosome. That tells you that this is coming from a male. And notice also the difference between the X and the Y chromosome, right? Unlike in females where the X and X chromosomes are the same size, the Y chromosome is this little pathetic thing with only a few genes on it, right? There's even some debate about whether it's slowly eroding away and will disappear completely. But notice that they wouldn't be able to actually form an actual X if they wanted to, right? So there's no centromere between them. So, and you can see the variation in the chromosome sizes. Some of them have a lot of genes, some of them have less genes. One important thing to remember is human DNA, along with DNA of most things, there's a lot of junk in there. Just stuff that is just built up over time that doesn't necessarily code for a protein, and it's not a functional gene, right? Might have some other functions possibly that we don't know about, but a good chunk of it is what we call quote-unquote junk DNA. Alright, so here's a process of sexual reproduction at its most basic, right? Here's an egg about to be penetrated by a sperm. So this is sexual reproduction, and these two things are gametes. So there's a difference between what happens with them versus mitosis. Mitosis applies to every kind of cell, but meiosis is specific to gametes in sexually reproductive organisms. So what is the difference between meiosis and mitosis? Well, there's a few differences. The biggest one is that goal of meiosis is not to simply duplicate the genetic material and then produce an exact clone copy of the cell. The goal of meiosis is not so much to copy the chromosomes, but to half the chromosomes. Why does this make sense? Well, because the point of sexual reproduction is that you half the chromosomes so that you can later recombine them with another set of chromosomes that are halved, and you end up with a full complement, right? And that is the most boring way to describe sex ever. So we have to distinguish between two sets of cells or organisms. The diploid, which is known as 2N, this is when you have the complete set of chromosomes, one from each parent. In the case of humans, we already showed you, there's 46 total chromosomes, and there are 23 pairs. Each pair, one comes from each parent. In contrast, the N version, the haploid version, is half of that. So gametes are sex cells, egg and sperm. They are haploid. They have half the chromosome set, right? And it's a toss-up. They could have some from mom, they could have some from dad, and it really depends on which chromosome it happens to be at any given time. And you'll find that that's a very important thing. that happens. So again, the goal of meiosis is the reduction in the number of chromosomes, not straight cloning. And you see differences between the three major groups of eukaryotes with regards to when you see haploid versus diploid stages. In the case of animals, most animals that are engaged in sexual reproduction, definitely, but most animals exhibit the diploid condition through life. As a multicellular organism, they are diploid. They have two sets of chromosomes. Have a chromosome pair. And the only thing that is haploid where meiosis occurs is in specialized sex cells, the egg, the sperm. The rest of the body is undergoing mitosis all the time. So we say that the diploid condition is the typical condition for an animal. On the flip side of that, fungi are essentially the opposite of that, because fungi spend most of their time in the haploid condition. So if you look at a mushroom or a bracket fungi, like one of those shelves that are on a tree, or any other kind of fungus, what you see is that most of the time the fungus that you see is going to be a haploid. And that haploid, the haploid cells in that mushroom are undergoing mitosis. And when they undergo mitosis, they just produce another haploid and another haploid. Right? They double their DNA and then they half it. Double DNA and half it. So they're undergoing mitosis with haploid cells, unlike animals which undergo mitosis with diploid cells. The only time you see a diploid fungus is when they engage in sexual reproduction and the gametes come together. And when that happens, you have a zygote, which is a temporary condition right after fertilization. That's diploid. And then what's going to happen is as it becomes an adult, it will undergo meiosis again, and it will produce that haploid condition. And that haploid condition will continue. Now, plants are in between. And depending on the kind of plant you're talking about, you can see it being predominantly haploid. And that's when we talk about they have their gametophyte stage. Or they could be predominantly diploid and undergoing their sporophyte stage. So it's quite interesting, really, depending on the type of plant. So usually we associate sort of more primitive plant, not that I like that word, but more primitive plants tend to be the more haploid version, and the more later evolving plants, your flowering plants, for example, exhibiting the more diploid version, right? But there are many, many exceptions to that rule. So you could basically argue that mitosis and meiosis are occurring in these things, and depending on the stage, the plant can be haploid or diploid. So it's really cool. And we call this the alternation of generations, or we say they have alternating generations. So it's very neat. So remember, the animal version doesn't always apply. We like to think it does, but it doesn't. But all these are answers to dealing with being alive and creating new cells as you grow and as you adapt to your environment. So quick reminder, how many chromosomes do human cells have? Well, typical somatic or body cells will have the 46 chromosomes total, 23 pairs, 23 pairs, right? And that includes a pair of sex cells, or sex chromosomes. Now, how many chromosomes do human gametes have? Well, if there's 46 in the diploid, then there's going to be 23 in the haploid, right? So, again, just driving the same point over and over and over to make sure. Homologous chromosome pair, the maternal side, the paternal side, each of those is going to undergo replication, and you're going to end up with paternal chromosome and a maternal chromosome now looking like the X with sister chromatids, right? Each of them going to be clones, and they're going to be held together by a centromere. So now that we know that, let's talk about how these things organize. So in a typical cell here, we have another cell. It's got 6 chromosomes, right? So the diploid number is going to be 6 because there's 6 X's floating around there. But of those X's, you see some of them are red, some of them are blue. Half of them are red. That'll be the maternal set. half of them are blue, that is a paternal set. If the diploid number is 6, then by default, the haploid number is 3. So that means that the gametes that are going to be produced as a result of this are each going to have 3 chromosomes at the end. Which 3? Who knows? It's like the lottery. So again, meiosis has a different goal than mitosis. The goal of meiosis is to produce haploid gametes, and you're not worried about them staying haploid for long because they're undergoing sexual reproduction and they're going to then recombine with another individual's gametes, and you'll end up with that diploid condition. So don't worry about that. So what's the benefit of all this? Well, simple. You're going to recombine with some other cells. And anytime you recombine, you're introducing a very important and very wonderful fundamental thing that evolution needs to drive forward, and that is called variation. Right? So the big benefit is just that. It's a faster way to have variation. And as soon as sexual reproduction evolved, it took off because all of a sudden evolution can work in overtime because now there were all this new variation to work with and to mold and sculpt and create with. So it's quite a wonderful thing. Who'd have thought? Okay, so I'm slowly building to the big picture here. So again, you're seeing chromosomes. These are the homologous pair, one from mom, one from dad, the red and blue, just like the previous example. They replicate, they produce their copy, so now we have the sister chromatids, the X's. Now we're going to undergo 2 different things. The first is meiosis 1. Meiosis 1 is where you're going to separate the chromosomes. You're going to separate the homologous chromosomes in the cells. And then meiosis 2, you are now going to separate the sister chromatids. So you start first with a cell that has diploid number with chromosomes that have been duplicated, and then right away you're going to cut at the end of meiosis 1, you're going to cut the number of chromosomes each cell has in half. And then meiosis 2 will then separate the chromatids, right, which are just copies of each other. So the easiest way to think of it is meiosis 1 is the big sort of thing that's different from mitosis. And meiosis 2 stage, which follows immediately after, is essentially kind of like mitosis. So unlike mitosis where you end up with 2 daughter cells, in the case of meiosis you end up with 4. And what do we label these things? Well, we label them the same thing that we labeled mitosis. So interphase is happening. It's happening, always. And then meiosis 1 happens. So in meiosis 1, you're trying to separate the chromosomes. You're trying to half that number. You're trying to go from diploid to haploid. So how do you do this? Well, first things first, your sister chromatids are together, but they're still going to line up as homologous chromosomes. And you're going to form essentially a 2X pair for each type of chromosome from mom and dad. And we call this the tetrad. And on top of that, not only are they tetrad, they're lined up beside each other, but they're also touching. And where they touch, that's called the chiasmata. It becomes important later. The chiasmata is this place where you can potentially have genetic material exchange between the 2 chromosomes. And when that happens, that introduces even more variation to the system. So now what are we going to do? It's prophase 1, and we use prophase 1, metaphase 1 to distinguish meiosis 1 from meiosis 2. Prophase 1 is the same as mitosis, except this time you're forming these tetrads, and they cross over at the chiasmata. Now you're entering to metaphase. Again, metaphase is important. Metaphase 1, you're lining up the tetrad on the metaphase plate. So unlike in mitosis, where in metaphase of mitosis you lined up all of the chromosomes in the middle and you had 6 of them going across the whole length of the cell, you now have 6 of them, but they're paired up. So all you're doing is lining up 3 tetrads in the middle. All right? So that's key, because then you can distinguish this from mitosis. Then what happens? Well, anaphase happens. They split, right? They've attached to those microtubules in the kinetochore. Now they've split, and each X is going to its respective pole. right? One's going to the North Pole, one's going to the South Pole. The sister chromatids are still there, right? You still have an X. Sister chromatids is still there. When they pull apart, notice on this picture, some of the red is on the blue chromosome and some of the blue is on the red chromosome. That's an example where they crossed over and they switched genetic material. So that's the end of meiosis 1. What happens next? Well, now you have 2 cells, right? So you're undergoing telophase 1, cytokinesis, and you're producing 2 daughter cells, but it's temporary. And now they're going to enter into meiosis 2. In meiosis 2, they're now going to do essentially what mitosis does, except that they have half as many cells, or half as many chromosomes to do it with. So they're going to do the same thing. Prophase 2, they're going to start lining up, and everything's going to start getting ready in each cell, right? You now have 2, so you're following 2 at the same time. Metaphase 2, they line up on the metaphase plate. Instead of having one cell with 6 chromosomes lined across it, or one cell with 3 tetrads lined across it, you now have 2 cells, each of which have 3 X's lined in the middle on the metaphase plate. So this is metaphase 2. Again, learn it, how to draw it. Then it's going to split, anaphase 2, and then telekinesis and cytokinesis is going to happen. Now you end up with 4 daughter cells. Each of them have half the genetic material of the original parent cell. And these 4 daughter cells are the gametes, the sex cells. They can be 4 sperm cells, or they could be 4 egg cells, although in most vertebrates you only produce 1 viable egg cell, and the rest become little structures called polar bodies. But nonetheless, you produce 4 cells. That's the bottom line. Okay, so how does the process of meiosis introduce variation? Well, there's 2 main ways. The first is through the process of independent assortment. By splitting things up and not really knowing if you're going to get mom's version of a chromosome or dad's version of a chromosome, well, you get a random assortment of genes, and anything is possible. So here are 2 chromosomes, and one is the maternal one is the paternal. 2 genes are identified here: eye color gene, hair color gene on each of the chromosome. They each code for their respective color. Okay, wonderful. The law of independent assortment is saying that the chromosomes will split however, and there's no rhyme or reason to it, it's just random. So if you have a situation like this with those same chromosomes and they split, well, you end up with 4 daughter cells at the end of meiosis. What combination could you get? Might you be a black-haired, brown-eyed girl? Are you going to be a red-haired boy with blue eyes? Are you going to be a red-haired boy with brown eyes? Are you going to be a black-haired kid with blue eyes? Right? Any combination of these is possible. Multiply that by how many chromosomes you have, and you start to see that you can get anything, right? And again, these are haploid, so they're going to refuse during fertilization and get even more variation. So what ends up happening is you can produce offspring that are quite different from the parent. The wonderful thing. So independent assortment, it's not just a good idea, it's the law. Now the other thing we already talked about a little bit is when they form these tetrads, right? They pair up, and that process of pairing up is called the synapsis. But the crossing over point is the chiasmata. And when that happens, they're exchanging material. So anytime this happens, you end up with a chromosome that has some extra genes from another chromosome that it lined up with, right? So some of mom's genes like rub off on the dad chromosome and vice versa. We call these recombinant chromosomes, right? If you think about recombinant technology, in terms of biotechnology, all that is going in and manipulating the genetic code by adding or subtracting things, right? By gene editing, and we produce recombinant DNA. It's the same thing. You're recombining it, right? You're combining it and producing a different version. Slightly different in this case. So crossing over. So independent assortment, crossing over, 2 major sources of variation in sexual reproduction as a result of meiosis. So just in recap, meiosis has 3 events that make it unique. The first is that process of synapsis and crossing over that you see in prophase 1 with the tetrads. The tetrads themselves being present and forming together, that's a unique meiosis event that you don't see in mitosis. And during anaphase 1, when things split, that first stage of mitosis, you're actually reducing the number of chromosomes. So you're actually separating the chromosomes and not just the sister chromatids. So those three things make it separate from mitosis. Mitosis doesn't do any of this stuff. So here's a side-by-side comparison again. Notice the metaphase, right? You have the cell, same amount of information, same number of chromosomes. In the case of meiosis, they form these tetrads in prophase. They're just floating around in prophase of mitosis. Then in metaphase, you see how the mitosis ones line up, those 6, versus the meiosis ones, those 6, how they line up, right? And they can line up any way, right? It's not all the parental ones have to be on one side and the maternal ones have to be on the other side. That's the law of independent assortment. And then when separation happens, you get some crossing over, right? You get some recombinant chromosomes. And then by the end of it, the daughter cells are going to have half the amount of genetic material as compared to the daughter cells of mitosis that are just smaller versions of the parent cell. So a side-by-side comparison, the best thing to do is just keep drawing these things and learning them that way. Okay, so why does all of this stuff matter? Well, there's a lot of reasons it matters. Obviously, it's an ultra vital part of biology, sexual reproduction. It could be traced to a lot of diseases, but to give you a more ecological example, of course, higher temperatures are a big thing, especially for climate change. And one thing people have noticed is for a lot of plants, temperature can be really important in maintaining efficient meiosis and reducing the amount of failed meiosis or meiotic failure, basically, which often results in inviable gametes or in inviable organisms potentially. So this is just some area of research that's still pretty modern, but there seems to be a pretty strong relationship between temperatures, body temperatures in both plants as well as in mammalian cells under certain conditions, and the likelihood of error during meiosis. Right, and we'll talk a little bit more about those type of errors later. Sometimes you get unequal division, stuff like that. So there is the potential, certainly other potential from chemicals and things like that to interfere with the process of meiosis. And if that gets interfered with, well, you're talking about interfering with reproduction, which is going to interfere with populations, which is going to interfere with survivability. So very important stuff. So I hope you enjoyed this little discussion about cell division. I think it's an absolutely fascinating topic. And this is Dr. F, over and out.